Hello everyone, welcome back to Nucleotides Biology Discuss. Today we have to continue our paper Genetics and this is lecture number 21 and this is for +3 third semester student. Today we have to discuss about the types of crossing over and what are the factors that affect the process of crossing over. Okay, so let's discuss about the types of crossing over first, then we'll discuss about the factors affecting the process of crossing over. Okay, so let's start. Crossing over, that is divided into three types. Crossing over, that is having three types. On the basis of what? On the basis of the number of chiasmata. Number of chiasmata formed. Okay. How many chiasmata that is formed during the time of crossing over? On the basis of that, we can divide the crossing over into three types. What are these? The first one, single crossing over. The second one, double crossing over. And the third one, this is multiple crossing over. Okay, we'll discuss this one by one. So first one, single crossing over. From this name, we can just clear that single crossing over that deals with the only single number of chiasmata will form between the non-sister chromatid of the homologous chromosome. Okay, so what will happen here? Suppose this is one chromosome and the homologous partner of this chromosome, suppose this one, and here the crossing over takes place only at one point. Okay, so here this one, that is the point of crossing over, and that is known as what? Chiasmata. How many chiasmata that is formed here? Only single chiasmata is formed here. So that this one, that is called as single crossing over. Okay, so one chiasmata, only one chiasmata will form between the non-sister chromatids. Okay, we have already completed our crossing over, the mechanism of crossing over that we have discussed earlier. We have not yet attended that class. You can go to our playlist and there is presence of that lectures. You can enjoy that. Okay, so let's continue to the second type, that is the double crossing over. Okay, yes, from the name it is having two chiasmata. Double crossing over. Okay, two number of chiasmata that is formed during the time of crossing over. Suppose this is one chromosome and this is another homologous chromosome, suppose, and here between the non-sister chromatids, suppose there is formation of two chiasmata, then that is known as double crossing over. Simple. Okay, then let's move to the third one, that is multiple crossing over. From the name it is suggesting that there will be formation of more than two number of chiasmata, that is known as multiple crossing over. Okay, in multiple crossing over there will be formation of more than two number of chiasmata. Okay, suppose this is one chromosome and its homologous partner suppose come here, and here they are come in contact during the time of crossing over. Okay, who can say? The process of movement of the homologous chromosome towards each other, that is known as what? Synapsis. And this complex, that is your synaptonemal complex, and in that synaptonemal complex, if you see properly, here this is one chiasmata, here this is another chiasmata, and here this is another chiasmata. How many chiasmata is formed between these two chromosomes? Three chiasmata. Then that will be called as what? That will be your multiple crossing over. Okay, so this is all about the types of crossing over. let's come to the main point, that is factor affecting the process of crossing over. Factors affecting the process of crossing over. Okay, first of all, the crossing over purely depend upon the distance between the gene. First one, that is distance between the gene. We have discussed about this. If the distance between the gene that is more, then the rate of crossing over that will be also more. If the distance between the gene is less, then the rate of or the percentage of crossing over will be also less. That means this one that is directly proportional. This one that is directly proportional to the rate of crossing over. Okay, so distance, if you consider the distance between the gene that are directly proportional to the crossing over. Okay, if you see two chromosome or two genes of the homologous chromosome, and if they are separate at more distance, okay, suppose the gene A and here this is gene B, suppose they are present at a distance, then they have chances of crossing over at this point. There is chances of crossing over between at this point, at this point, at this point. So the more chances of crossing over will be there if they are at more distance. If they are very close together, then there will be less distance, or if there will be less distance between the genes, then there will be less chances or less area for the crossing over. Okay, so for this concept we have also discussed about this during the time of the what is the relationship between the linkage, crossing over, and the distance between the gene. In the last class we have discussed about this. You can go to that playlist and you can see the our genetics lecture number 20. Okay, so this one first factor, that is distance between the gene. The second factor, that is the temperature. Okay, the temperature that is also directly proportional to the process of crossing over. If the temperature that become very low or temperature become very high, temperature if that become very low or temperature become very high, then that will promote the process of crossing over. Okay, if we take some chromosome or if you see some organism, in that organism during the time of the process of gamete formation, if you provide more temperature, then there is chances of more crossing over. Or if you provide very less temperature, there is also more chances of the crossing over. So that this is also directly proportional. Temperature that is also directly proportional to the crossing over process percentage. Okay, so very low temperature or very high temperature that promote the process of crossing over. The third one, that is age of the individual. If you see the age that is indirectly proportional. Suppose the individual having more age, then there will be less chances of the crossing over. So that what happened, if you see the crossing over, that is the normal process for an individual during the time of gamete formation. If any individual that become very older and older, and at old age they are becoming the parent, if they become parent at old age, then there is very chances that that person may become having some kind of defect in gametes, so that in future the offspring will produce, that offspring will have some defects in them. Okay, so that age is indirectly proportional to the crossing over. If age increase, then there will be decrease in the percentage of crossing over. Okay, Then the fourth factor, that is sex of the individual. Okay, in case of sex, we have two sex, one is male, another one female. If you see, in case of female individual, there is presence of, suppose in case of human being, there is presence of how many chromosome? 46, and they have two sex chromosome, and both are X chromosome. But in case of male individual, there is presence of X and Y. Okay, suppose in case of your male, we have X and Y, and in case of female, we have X and X, and these two are homologous. These two are homologous. But X and Y, these are non-homologous. These are not homologous, non-homologous. Okay, the size of the X chromosome, and if you compare with this Y chromosome, the Y chromosome is very smaller size. The number of genes which are present over X chromosome, that is very high as compared to the Y chromosome. Okay, the position of the centromere in X and Y chromosome, that is also different in both X and Y chromosome, so that they are not the homologous chromosome, they are non-homologous chromosome. And crossing over, that occurs between only homologous chromosome. As here, all the chromosomes which are in pair condition 1 1 2 2 3 3, and if you comes to X and X, all chromosomes that are homologous, then there will be more chances between this X 1 and 1, 2 and 2, 3 and 3, and X and X. But here we have 1 1 2 2 up to, if you see, X and Y. Okay, here crossing over is chances, here crossing over will occur, but in between this X and Y chromosome, there will be no crossing over. So that there is more chances of crossing over in case of female individual than the male individual. Okay, so male have more chances of female have more chances of crossing over. Okay, then let's see several chemicals that also affect the process of crossing over. Factor number 5, there are certain chemicals, okay, some chemicals that affect the process of crossing over. So for example, that is actinomycin. Okay, that we use this as antibiotics. So some chemicals that affect the crossing over process. Okay, for example, that is your actinomycin. Actinomycin. Okay, or mitomycin that actually promote the mitosis process. Okay, so these actinomycin, mitomycin, they promote the crossing over process. They promote crossing over. That means if we take actinomycin or mitomycin during the time of gamete formation, then there will be chances of more crossing over. If there is more crossing over, in the next generation we will get more recombination. The new individual will have more recombinant characteristic features. But if we are not taking this antibiotics, then there is less chances of crossing over. That means there will be more linkage. Okay, so that this is the chemicals. And sixth one, that is the number of chiasmata. Yes, that is also another factor, chiasmata. Okay, if there will be more chiasmata formation, then there will be more crossing over. Okay, and we will discuss another concept in future, that is interference. What is that interference? If you see crossing over between the two genes, one number of crossing over that may interfere the other type of crossing over, that we will discuss. That is known as what? That is your interference. Okay, that we will discuss later. Then the seventh one, that is the nutritional factor. Nutritional factors. What is nutritional factor? Some of the nutritive substances, for example, calcium or magnesium, they also affect the process of crossing over. For example, in Drosophila, there is high or there will be, if you put the Drosophila in high concentration of calcium, then there will be less chances of crossing over. That means that calcium, if you take more calcium, then there will be less chances of crossing over. Okay, so some nutritive substances that, for example, calcium, or that may be magnesium, that may affect the process of the crossing over. For example, in case of your Drosophila, if the Drosophila grow in high calcium concentration, high calcium concentration, then what happen? There will be less crossing over, or the crossing over percentage will decrease with increasing in the amount of the calcium, that is the nutritive substance. Okay, then the eighth one, or this one, that will be the last one for the factor affecting the process of crossing over, that is position of the centromere. Position of centromere. Okay, if you see the centromere, if the genes which are going for the crossing over, or in between the two genes, if there is occurrence of the crossing over and that genes are close to the centromere, then there will be less chances for the crossing over. Suppose this is one chromosome and this is another chromosome. If suppose the genes are located nearer to the centromere, then there is less chances of crossing over, because if there will be more distance between the genes, then there will be more chances of crossing over. And if they are closer to the centromere, that means they are very close, or they are having the very less distance, so that there will be chances of less crossing over. Okay, so if the genes are closer to the centromere, closer to centromere, then there will be less chances of crossing over. Okay, so these are the factors which affect the process of crossing over. Okay, generally crossing over occurs between the homologous chromosome and between the non-sister chromatid of the homologous chromosome. And at what stage or in which time the crossing over occurs? When the particular individual is going to produce the gametes, at that time crossing over occurs. And during the time of gamete formation, which cell division occurs? That is your meiosis occurs. And during the time of meiosis, if you see in which stage the crossing over occurs, that is in meiosis 1. But in meiosis 1, there is several steps of karyokinesis 1 and cytokinesis 1. And the crossing over occurred during the time of karyokinesis 1. And in karyokinesis 1, there is prophase 1, then your metaphase 1, anaphase 1, and telophase 1. In which stage the crossing over occurs? That is in prophase 1. And if you see the prophase 1 in more details, that is having several 5 steps. The first one, lactotin, zygotin, pachitin, diplotin, and dikinesis. The crossing over, the time for the crossing over, or the phase at which the crossing over occurs, that is your pachitin stage. But crossing over start at which phase? At which stage? That is your zygotin stage. That means in the zygotin stage, what happens? The homologous chromosome, they come close towards each other, and that movements of the chromosome, that is known as for synapses. Okay, and after synapses, there will be exchange of the chromosomal segment at the Pachitin stage. But after that, at diplotin stage, both the homologous chromosomes, they start separating from each other. And during the time of the separation of the homologous chromosome, there will be occurrence of a X point, that is known as what? Chiasmata. Okay, and on the basis of number of chitomata formed between the homologous chromosome, we have divided the crossing over into three types. The first one, single crossing over, if there is formation of only one chitomata. Second one, double crossing over, if the crossing over occur at 2 points, or there will be formation of two number of chitomata between the non-sister chromatid of the homologous chromosome. And if there will be formation of more than two chiasmata between the non-sister chromatid of the homologous chromosome, then that will be called as multiple crossing. Okay, so this is enough for this class. In the next class we'll continue from the types of linkage, and in that part we will discuss about the complete and incomplete linkage, and in that incomplete linkage and complete linkage we will discuss about the one of the experiment that is performed by Bartison and Punet by taking the Lathyrus odoratus, and in that we will discuss about another concept, that is your gametic coupling concept and gametic repulsion concept. Okay, I hope you may enjoy this lecture. If any doubt, you can write in the comment section. Please like, share and subscribe to our channel. Thank you.